The structure of DNA. The structure of DNA was first discovered in 1953 by Watson, Crick and Wilkins with the help of Rosalind Franklin. In 1962 they were awarded the Nobel Prize for their work. DNA stands for deoxyribonucleic acid. It is composed of repeating units called nucleotides. Each nucleotide is composed of a phosphate, a deoxyribose sugar, and a nitrogenous base. The nucleotides are linked together so that there is a phosphate and a sugar in the backbone with nitrogenous bases protruding. There are 4 types of nitrogenous bases: adenine, thymine, cytosine, and guanine. But this is now only half the DNA molecule. Opposing this is a mirror image type molecule, except this time the sugars are facing the other direction. There is still the backbone of phosphate and sugar, but now the nitrogenous bases are complementary to the bases on the other strand. Complementary means matching to complete the set. Thymine is complementary to adenine, adenine is complementary to thymine, guanine is complementary to cytosine, and cytosine is complementary to guanine. These are called the base pair rulings. There are hydrogen bonds between the complementary bases. There are 2 hydrogen bonds between adenine and thymine, whilst there are 3 hydrogen bonds between cytosine and guanine. The DNA molecule is described as a twisted ladder, with the phosphate and sugar backbones as the legs and the nitrogenous base pairs as the rungs. You will notice that the legs in the ladder run in opposite directions. We call this anti-parallel. It is the sequence of the nitrogenous bases on the coding strand that makes up the genetic code, and it is this sequence that codes for all protein production in cells. In the S stage of interphase, the DNA is copied, turning it from a single-stranded molecule into a double-stranded one. This process is called DNA replication and happens in a number of steps. Firstly, the hydrogen bonds between the complementary base pairs are broken by an enzyme called DNA helicase. Now the nitrogenous bases on each side of the ladder are exposed. Next, another enzyme called DNA polymerase puts the new complementary nucleotides on the exposed nitrogenous bases according to the base pair rulings. If there is an adenine exposed, then it puts a thymine in its place, and so on. Then the enzyme DNA ligase comes along to tie the nucleotides together along the backbone. The result is 2 identical strands. DNA replication is said to be semi-conservative because each strand is composed of half old DNA and half newly synthesized DNA. Thank you.